Hello everyone and welcome to the 7th student. Today we're looking at C2.1, which is chemical signaling in IB biology. So what is chemical signaling? Okay, it's a way for cells of the body to communicate with each other and coordinate action, right? So cells communicate by sending and receiving signals. These can be chemical signals, right? So a chemical substance used to send a signal is called a ligand. So for example, here in pink, you see a type of ligand. And then we have receptors, which are proteins, where the ligand can bind. And that stimulates a response by the cell, which has the receptor. So this binding is specific, and so both the ligand and receptor bind, but then are unchanged by the binding. That means the ligand can then go ahead and bind to another receptor. This specificity is very, very important because, as you can see here, in the case of a pancreatic cell, if we're releasing insulin, if insulin is the ligand, we want insulin to bind to liver cells so that they will uptake glucose, but not necessarily neurons, right? So here you can see that specificity in action. That allows for very specific and tailored communication. Let's look at an example of why this communication is important. So quorum sensing, you probably have never heard that word. Quorum sensing happens in bacteria, and it's basically the fact that bacteria have some behaviors that only happen when the population density rises above a certain threshold. So let's look at this case. Vibrio fischeri are a type of bacteria, and they secrete signaling molecules, right, that bind to receptors on the same cell. So basically, Vibrio fischeri are secreting molecules that bind to receptors on themselves. When there is sufficient binding, and for that, you need enough bacteria secreting ligands, gene expression is changed, causing a change in activities. More specifically here, the signaling molecule binds to a receptor, and that causes a change in the DNA here, right? Causing expression of luciferase, which is an enzyme that catalyzes a reaction that produces light. And you can see Vibrio fischeri associate with some animals, and you can see here the luminescence produced by these bacteria. So importantly, right, this signaling allows the bacteria to detect whether there are enough bacteria, right? Whether the density of the population is enough. This is a great example of how communication can trigger action. So how do we communicate? What type of signaling chemicals are there? So we can categorize them into hormones, first of all. So hormones are produced by glands in the body, and they are transported by the bloodstream to all parts of the body. They only have an effect on target cells, which have the receptors for the hormone, and the effects can last for hours. So examples of this are testosterone or insulin. Then we have neurotransmitters. So these are chemicals that transmit signals across synapses when a nerve impulse reaches the end of a presynaptic neuron. So they move a very, very short distance, and they convey the signal much quicker than hormones do. But the effect lasts very, very little time. Examples of this are acetylcholine or dopamine. Then we have cytokines, which are not transported as far as hormones, but they act in a similar manner. So they normally just act nearby from where they are secreted. And again, they bind to receptors in the plasma membrane. So they are very, very similar. One big difference is that cytokines can normally bind to several types of receptor. They're not as specific. They play a huge role in the immune system, and examples are interferon and interleukin. And then finally, calcium ions are used as signaling chemicals in muscle fibers and neurons. So in muscle fibers, they unblock muscle contraction, and in neurons, they allow for membrane potential to change and for neurotransmitters to be secreted. But we're going to focus on hormones and neurotransmitters. So first, it's important for you to know that they are very, very diverse chemically speaking. So that makes sense because there are a large amount of signals that need to be conveyed, and because these chemicals are specific to a receptor, we need tons of them for every signal that needs to be given. So starting off with hormones, they can be steroids such as testosterone or estradiol. They can also be amines, and they can be peptides as well, right? So for example, an example for hormone that's an amine is melatonin, and a peptide is insulin. And then for neurotransmitters, they can also be amines, so dopamine, for example. They can also be peptides, such as endorphins. They can also be gases, such as nitrous oxide, and they can be esters, such as acetylcholine. So a huge variety here. We also need to distinguish between hormones and neurotransmitters in terms of the effect they have, right? I talked about this briefly, I think, but for example, neurotransmitters are transported only a very, very short distance across the synaptic gap between two neurons. So that's normally 20 nanometers, tiny, tiny, tiny. Whereas hormones can be transported from the gland that produces them to all parts of the body. So, for example, the pituitary gland, which is right next to the brain, produces LH, which then goes to the testes, right? So it has to travel a really, really long way, and that's why they go in the bloodstream. Great. So then what happens when a ligand binds a receptor? This can happen in several ways. So firstly, some signaling chemicals enter the cytoplasm, whereas others do not. This depends on whether the ligand itself has the ability to go into the cytoplasm or not. Remember, the plasma membrane is only permeable to some molecules. Therefore, we have intracellular receptors for those that can enter the cytoplasm. And these intracellular receptors, as you can imagine, are going to have hydrophilic amino acids on their surface because that way they will remain dissolved in the nucleus, which is a water solution. And then the receptors for chemicals that do not enter are located in the plasma membrane. These are called transmembrane receptors, and they will have hydrophobic amino acids on their surface where they touch the phospholipids, right? And then hydrophilic amino acids on the surfaces where they face the inside and outside of the cell. So remember the hydrophilic versus hydrophobic amino acids and where they're placed, because that will come up in exams. So what actually happens when the ligand binds a receptor? Well, by itself, nothing, right? So receptors have to initiate what's called a signal transduction pathway, which just means turning the signal binding into action. So transmembrane and intracellular receptors use slightly different transduction pathways. So transmembrane receptors, when the ligand binds, it normally changes the structure of the receptor. So the inner side, the one facing the cytoplasm, can activate a secondary messenger, which then carries out the response, normally gene expression. Intracellularly, the ligand and the receptor normally form a pair. They form a ligand-receptor pair, which can normally go into the nucleus and bind DNA directly to regulate gene expression. So examples of hormones that work this way are steroid hormones. Because they are hydrophobic and very, very small, they can enter the cytoplasm. And examples include testosterone, estradiol, and progesterone. So a little more about these. Estradiol and progesterone are involved in reproduction. And you do need to remember this, okay? So estradiol targets cells in the hypothalamus, causing them to secrete gonadotropin-releasing hormone, or GnRH for short. So this hormone then triggers the release of LH and FSH. Okay, I know there's a lot to remember here. Estradiol can induce production of GnRH, and it can also inhibit it at different points of the menstrual cycle. On the other hand, progesterone is produced by the ovary, and it targets cells in the endometrium, inducing cell proliferation to maintain the endometrial lining, and that's in order to support the developing fetus. So take notes on that, remember it, because it will come up, and it'll be a pain if you don't remember. So now let's look at a bunch of examples of ligand receptors and what happens. So acetylcholine, right? Acetylcholine is a type of neurotransmitter. Neurotransmitters get released into the synaptic gap and then diffuse to the membrane of the postsynaptic neuron. Here, they're going to bind to transmembrane receptors, and this will cause the membrane channels to open and ions to move into the postsynaptic neuron. That changes the membrane potential and starts a nerve impulse. We'll look at that in more detail in the next topic, but this is all you need to know for now. Then we have GPCRs. GPCRs are super important. Actually, I can't remember the number, but about 70% of drugs target GPCR. So they're very, very important in medicine. A lot of diseases are caused by inefficiencies in GPCRs. So how do they work? Okay, so GPCRs are basically a very large and diverse group of transmembrane receptors. They have a curious shape. They have like 7 stalks that go through the membrane, as you can see here. And basically, they transduce signals using a second protein, which is called the G protein. The G protein is this in green. It has 3 subunits, the alpha, beta, and gamma subunit. So the alpha is kept inactive by a GDP that's attached to it. But when a ligand binds, it changes the shape of the receptor, causing the GDP to detach. And that allows GTP to bind instead. That activates the G protein and splits the subunits. So alpha splits from beta and gamma. And then alpha can then bind to an effector protein and have an effect further down the line. Let's look at an example of that. So epinephrine. Epinephrine, or you probably have also heard of it as adrenaline, binds to a GPCR. And what happens, as we just said, is the alpha detaches from the beta and gamma due to that GTP. The alpha then binds to adenylyl cyclase, which adenylyl cyclase is another GPCR, actually. And what that's going to do is it's going to convert ATP to cAMP. This is the secondary messenger that we were talking about. And that eventually, has further effects, which causes a cellular response. In this case, the conversion of glycogen to glucose, which is then released into the blood in preparation for intense effort, right? Because that's why adrenaline was secreted in the first place. This is complex, I understand that. Take a while to look at it, maybe draw it yourself so that it really sinks in. Another example, tyrosine kinases. So a kinase is just an enzyme that adds a phosphate group from ATP to another molecule. So tyrosine kinases, more specifically, add a phosphate from ATP to a protein, and specifically to the tyrosine in that protein. A tyrosine is an amino acid, okay? So for example, the insulin receptor works that way, okay? So the tails of the receptor in the insulin receptor are tyrosine kinases. So when the ligand binds, when insulin binds, these tyrosine kinases are going to connect. So the receptor changes shape, these connect together, and they phosphorylate each other. And that causes signal transduction. And eventually what this leads to is the movement of vesicles with glucose transporters to the plasma membrane so that glucose can enter away from the blood and into cells, which is the whole point of insulin, okay? And then finally, how are cell signaling pathways regulated, right? Because as we said, ligand and receptors are not changed permanently by binding. So technically, these pathways could go on forever and ever, but they need to be stopped and regulated. So first, we have positive feedback processes. So in positive feedback, the end product of a pathway amplifies the starting point. So more product is created, right? So it self-amplifies. So an example is in muscle cells, the ER, the endoplasmic reticulum, stores calcium. When a molecule called IP3 binds to its receptor, it causes release of calcium from the ER. That calcium activates the IP3 receptors even more, causing more release of calcium. So you see how it self-amplifies. However, in a negative feedback process, the end product of a pathway shuts off the start of the pathway. So an example is GnRH, right? Gonadotropin releasing hormone, which we talked about before. So it acts on the hypothalamus, inducing the release of LH and FSH, and that increases the release of testosterone. But testosterone then acts on the hypothalamus as well to stop the release of GnRH. So it's a loop, and it makes sure that not too much is being produced, okay? I know this topic is quite tricky, okay? So maybe watch these slides again if it's not clear. But we're going to move on to do some questions for a recap. So first, bioluminescent bacteria in the sea use quorum sensing to regulate light production. Which of the following statements about this process are correct? Quorum sensing relies on the secretion and detection of signaling molecules. Gene expression is regulated in response to population density. And bioluminescence in bacteria is an example of negative feedback. Okay, I'll let you think, but pause now, and I'm going to count down from 3, 2, and 1. And the answer is B, okay? I think this is quite straightforward. So remember, it's the bacteria releasing ligands that bind to receptors on themselves, basically, right? So they detect it themselves. It does affect gene expression, right? So the more population density, the more ligands are being produced, the more the receptors are being bound, and the more those genes are expressed through the transduction pathway. However, it's not negative feedback. In fact, it's positive feedback, right? Because the more population density there is, the more ligands they produce, so the more receptors they bind, so the more gene expression there is, okay? So it's B. The next question, which of the following hormones is lipid soluble and can pass directly through the cell membrane to bind to intracellular receptors? This is a memory game. Let's see you remember. 3, 2, and 1. It's estradiol, right? So remember, insulin, we just saw, binds to those tyrosine kinase receptors, which are transmembrane. Adrenaline, same thing. They bind to GPCRs, which are transmembrane. And acetylcholine is a neurotransmitter. So again, it binds to transmembrane receptors in the postsynaptic neuron. But estradiol, such as in the same way as testosterone, is a steroid hormone. So it can pass directly through the plasma membrane. Okay. And finally, what is the primary function of second messengers in hormone signaling? Again, 3, 2, and 1. Okay, so it's B. Why? Okay, A, they do not transport hormones through the bloodstream. That they do that themselves. The hormones just travel on their own. C is incorrect because hormones, lipid-soluble hormones such as steroids, they don't need second messengers to cross membranes. They diffuse freely. And D, they don't break down neurotransmitters. That is done, but it's done by enzymes, not second messengers. However, they do take part in the signal transduction pathway. That is, they amplify the signal inside a cell after a hormone binds to a receptor, leading to a larger response. Okay? I think that's quite straightforward. I know this concept in general, this topic is really, really tricky. If you have questions, please do leave them in the comments and I'll do my best to respond. And I'll see you next week for neural signaling, okay? Have a great week, everyone.